Today, red blood cell transfusions are given to patients with anemia who are unable to supply their body with adequate levels of oxygen. In 2017, transfusion medicine is undergoing a revolution. Drugs are now being linked to RBCs to achieve benefits that were not possible with traditional pharmaceuticals. Drug-linked RBCs are the next generation of blood transfusion. Normally, red blood cell transfusions are given to improve oxygen delivery to the body. Red blood cells circulate in blood vessels and release oxygen to the cells and tissues in the body using a molecule called hemoglobin. Today, the process of blood transfusion starts with collecting blood from a healthy donor. The blood is then processed to create a unit of RBCs, which is approximately 300 milliliters in volume. The blood is then stored in a blood bank refrigerator until needed. When a unit of RBCs is ordered, the product is released from the blood bank for a recipient, most likely a patient who is anemic. The product is then transfused. Drug-linked RBCs are the next generation of blood transfusion. In 2017, technology for linking drugs to RBCs is available, and the RBC is now classified as an advanced drug delivery system. RBCs can improve drug delivery in several ways. For instance, because the RBC can circulate for more than 100 days, it can increase the half-life of drugs. In addition, because RBCs remain within the vascular space, RBC-linked drugs will remain in the blood vessels. Additionally, because old RBCs are cleared from the circulation by macrophages in the liver and spleen, the RBC can be used to deliver drugs to these organs. This has tremendous significance for establishing immune tolerance to specific proteins. Being immune tolerant to a protein means that your body doesn't attack or form high titers of antibodies against the protein. If you want to remain healthy, being immune tolerant is important. So let's explore this concept in detail. Macrophages are a type of cell which regulate immune tolerance. To establish immune tolerance to a protein, macrophages must process the protein. Injecting a protein into the bloodstream is an inefficient method of delivery to macrophages. Because RBCs are naturally cleared by macrophages, proteins linked to RBCs are efficiently delivered to these cells. Macrophages engulf and digest old red blood cells. The proteins linked to red blood cells are then processed by the macrophage. Portions of the protein are then presented on the surface of the macrophage. Lymphocytes, which are the immune cells of the body, can then recognize the protein as a self-antigen and become tolerant to the protein. Simply put, protein linked to red blood cells is more efficiently delivered to macrophages compared to soluble protein. Therefore, protein-linked RBCs are being evaluated for clinical immune tolerance induction. Protein-linked RBCs are changing the process of blood transfusion. Blood will be collected from patients with autoimmune diseases. However, only small volumes of blood will be collected. Proteins will then be linked to RBCs using a method known as APEX. APEX is a convenient, universal, and safe method for attaching proteins to RBCs. First, the patient's RBCs are washed. Then a chemical powder, which is stored by the blood bank, is mixed with the RBCs. Finally, the protein-linked RBCs are washed to eliminate reaction byproducts. The protein-linked RBCs will then be transfused on the same day of blood collection, typically 1 to 2 hours after blood collection. The protein-linked RBCs will then establish protein-specific immune tolerance. Here is a comparison of blood transfusions for immune tolerance induction versus oxygenation. To create protein-linked RBCs, blood will be collected from the patient rather than a healthy donor. Small volumes of blood will be collected rather than large volumes. Blood will be transfused on the same day as collection rather than being stored. and blood centers will act as key sites for manufacturing and transfusing protein-linked RBCs. In summary, drug and protein-linked RBCs offer hope to patients with autoimmune diseases and are revolutionizing the field of transfusion medicine. At Biconcavity, we hope to improve the lives of patients with antibody-mediated conditions through education and the development of new therapies. Visit biconcavity.com for more information.